I'll try to make it as short as possible. First of all, in grade 12, this is the second topic, the topic of meiosis, and it carries quite a number of marks, so you need to be straight ahead with regards to it. You need not to just waste time on it. Yes, that's what I wanted to do. Excuse me for that. Now, the first thing that you need to know about meiosis is you need to be able to define what meiosis is, okay? You need to be able to define what meiosis is. Meiosis is what? Is a type of cell division that results in 4 daughter cells for each with half the number of chromosomes of the parent cell. Ithetha ukuthini ke lento? Ithetha ukuthi lento you are given kahle kahle there is a what? There is a cell. It's a pity at 9 to draw it here, but I will try to draw it here. There's this one cell, okay? Now this cell, we are saying it's going to do what? It's going to divide. How does it divide? It divides by this, okay? It divides first because in a division there's a 2. There's first meiotic division, there's also a second meiotic division. Then if first meiotic division, it gives rise to 2 daughter cells, okay? This is the first daughter cell, and there's the second daughter cell. Excuse my, let me undo here, okay? This is a second daughter cell. Now, ayiphelelanga meiosis lapha because it results in 4 daughter cells. That means after lo meiosis 1, kufuneka futhi again meiosis iphinde iqhubeleke and gives rise to the last stage of meiosis. So each of these 2 daughter cells is going to give rise to 2 daughter cells each, okay? It's going to give rise to 2 daughter cells each. So at the end of the process of meiosis, you're gonna have how many? You're gonna have 4 daughter cells. 1, 2, 3, and then the last one over there. Okay, this is the process of meiosis. Now, when you're looking at this, all right, these daughter cells, kufuneka zithini? 1, 2, 3, 4. They must not be the same as the parent cell. At the same time, nazo zoyi-4 kufuneka zingafani. How ukufana? That is, genetic material elapho ayizufana ne-genetic material elapha. Ingafani ne-genetic material lapha, iphinde ingafani ne-genetic material elapha. So all of them are going to be different from each other. This one will have its own, and then this one will have its own genetic material, and then again this one will have its own genetic material. Or let me rather do it like this, so that siyibonakalise ngokwe-colors. Then we just draw it kanje, and then we choose another color for the other one. This simply tells us what genetic makeup for all these daughter cells ayifani, okay? And also I would like us to have a different color for the parent cell, okay? Yes, so the parent cell was this color, and then gives rise to this one, and this one, and this one. So that's the process of meiosis. Ne? So in definition, it is as Lena Uibonayo, it's a cell division that results in 4 daughter cells, each with half the number of chromosomes of the parent cell. Now, the second point says, as in the production of gametes and plant spores. So as is in those 4, so it is here produced by 1, 2, 3, 4. Let me get my pen again. What are these 4 outcomes here? What are these 4 outcomes? If this is a human, it can either be an ovum, but this is oval. It can either be an ovum or it can be a spermatozoa. If it's what? It's a human. If it's not a human, it's a what? It's a plant. It's gonna be either ovules or it's gonna be a pollen grain, depending on the gender of the plant. That is to say, if the human is a male, le male, kuzophuma ntoni is spermatozoa. If yenzeka kwi-female, kuzophuma the ova, all right, amaqanda, ne? And then if yenzeka kwi-plant male, kuzophuma i-pollen grain. If yenzeka kwi-female plant, kuzophuma ntoni? I-ovules, all right? That's what all these things actually means. Now, moving on to the next slide, all right? So in the next slide, you are told about the things that you need to take care of, you need to pay a serious attention, which is a nucleus, a centrosome, and the cytoplasm, okay? Now, again, we'll need to highlight even here. So, you know the structure of the nucleus, but the nucleus contains, and it contains these parts. These are the parts of the what? Of the nucleus, all right? Let me also include the ribosomes there. These are part of the nucleus. Where is the nucleus? The nucleus is found in a cell, all right? This is a cell. The nucleus is found in a what? In a cell. This is a cell, okay? Now, as this is a cell, phakathi kwesele kukhona many organelles, but siphikisha i-nucleus kuba izosebenza ku-meiosis, ne? So ngaphakathi ke ngokwe-nucleus kukhona ntoni? Sizibalile endoplasmic reticulum abangaphandle nansi phaya kwe-nucleolus, ngaphakathi kukhona your nucleus, your nucleoplasm, your chromatin. Sihambele ke ngothina phaya kwintoni? Kula chromatin iphi? Ipha ngaphakathi. Kwenzeka ntoni kula chromatin? This is the chromatin network, nansi ijikeleze phaya all over the nucleus, ne? Kula chromatin network epha ngaphakathi sithi i-network i-chromatin kahle kahle, but njengoba kuthiwa network indlela eme ngayo kwawuphithana, it's a network, ne? It's networking, okay? So phakathi kukhona ke ngoku la chromatin network. La chromatin izoba matured, it's not yet matured then, that means izo-replicate, maturisha until ikhuphe ezi zinto sisibiza ngoba zintoni? Zii-chromosomes, all right? So i-cells end up ngosithi i-nucleus ayisena chromatin kuphela, it has got a what? The chromosomes, okay? I-chromosome number ke inside the nucleus iyadependa. It depends by which organism is that. Umzekelo, us as humans, we've got in each cell, in each nucleus, because nucleus is billions and billions of nucleus, but we've got nucleus nganye, we've got 46 number of chromosomes. 40, excuse my writing, 46 chromosomes. Omnye angayibhala as 46 chromosomes, angayibhala as 23 pairs, okay? I'm with 23, got 23 pairs of chromosomes. So it's not that, but it was 23, okay? The 46 chromosomes, but it's a ngokwe pairs. I-pairs ezihlele ngazo zii-pairs ezingaphi? Zii-pairs eziyi-23, okay? So these are the chromosomes. Now, you need to be able to draw and label the structure of a chromosome, okay? You need to be able to draw and label the structure of the chromosome. Here is the chromosome, but kahle kahle sizosibona i-structure of a chromosome in the next slide, all right? Here is the next slide, okay? Now, in this slide, we are seeing here that we're having a chromosome, but before we even go further, we must first define what chromosome is. Chromosomes are thread-like structures. This is isn't it? These thread-like structures are present in the nucleus, chromosome, which carries genetic information from one generation to another. So for you to be the way you are, it's because of your chromosomes that are genetic information. Excuse me, that are genetic information, taking genetic information from your parents. That means apha kukhona genetic material esuka kutata wakho kube khona genetic material esuka kubani. Oh, let me use another color then. Kube khona genetic material that is from your mother. All right. But here, as chromosomes on this side, they are not yet replicated. This cellular form. This is a chromatid. As is a fully matured chromosomes. Okay. But as chromatids, in as much as the chromatids, there's now information. Any is from paternal, which is father, and is from maternal, which is mother. Now, after in fact, stating with DNA, DNA replication, you know it. The outcome will now be a matured a matured chromosome that is having 2 chromatids. Okay, now the chromosome funeka ukwazi uyilabelisha. Okay, intoni le funeka uyibambile kule chromosome? Zii-parts zayo. One, there is this part that is in the center and this part that is in the center. All right, kula ndawo kudibana khona ezantonga zethu. Ezantonga besithe zii-chromatids, ne? So there's a chromatid from this side. This is a chromatid, there's a chromatid, and then this is another chromatid. Okay, all right. Now, as a chromatid ziyadibana pha esiphakathini. Yintoni lezi zidibana ngayo? Zidityaniswa isi-structure sesibiza kuba yintoni? Yi-centromere. Let me also put this one here so that uzokwazi ukuyibona. This is a what? A centromere, that one. Okay, it's not a centrosome. All right, it's a centromere. So le part idibanisa okanye phaa kudibana khona chromatids ze chromosome eyi-1 siyibiza ngoba yintoni? Yi-centromere. And then kukhona igama lithi centrosome. Ucentrosome xa sekusenzeka imeiosis, xa sekuphulishwa ezinye chromosomes zisiya kwenye pole, ezinye chromosomes zisiya kwipole, laa nto sithi yipole yiyona ntoni? Yiyona centrosome yethu. Okay, yiyona centrosome yethu. And then likhona ke ngone elinye igama eliconfusayo, ucentriole. Ucentriole phaya ngaphakathi kwi-centrosome kukhona structures that are there. These structures are known as what? They are known as centriole. So it's centrosome, centriole, centromere. Xa ubuzwa ku-exam uba kuthiwa label this part, you must never say centrosome, you must never say it's a centriole because you are going to lose that mark. Okay, so the next point is each chromosome is made up of DNA genes. It's made up of DNA genes. Baby, that's what my sister is made up of DNA genes that are tightly coiled many times around proteins. These proteins are called the histones that support its structure. Okay, so chromosome nganye is made up of DNA genes. Okay, all right. Now, I think, okay, the one other thing that we need to take a closer look is the issue of these terms. There is a term used here, homologous chromosomes. Okay, what is meant by homologous chromosomes? Okay, homologous chromosome, it is whereby a chromosome from the father part esuka kwicala katata, okay, idibana okanye ipherisha ne-chromosome esuka kwicala likamama. Okay, that means a chromosome ebiza ne-egg kamama, i-chromosome ebiza ne-spermatozoa sikabani? Sikatata. So xa zidibana ngenxa yoba ziphethe the same characteristic. Xa ndithi characteristic, umzekelo, kuba le chromosome njengoba sithe ine-genes, andisithe ine-genes i-chromosome, njengoba ine-genes, if le gene le chromosome kamama iphethe ii-genes zoba iba mngakanani, that means as a height, nalena kufuneka iphathe ii-genes ze-height. Okay, so xa ziphethe ii-genes ze-characteristic eyi-1, la height yi-characteristic, so it's the same characteristic, they pair together like that. And then sithi ngozenjani? Homologous chromosome. Homo means same, they carry the same characteristic. Okay, All right, so we know homologous chromosome. And then one other thing that we need to take a closer look at are these terms called sister chromatids. Xa kusithiwa sister chromatid kuthetha ukuthi wani? I-sister chromatid, njengoba i-chromosome A1 is made up of 2 chromatids. Eziya chromatids zombini ziku-chromosome A1. So these are sister chromatids ngale ndlela ne, zizalwa yi-chromosome A1, one chromosome, and then njengoba ziphaya zizi-sisters. So these are sister chromatids. Okay, so that's one term that we need to take a look at. So now I think we can move to the next slide. Okay, we can move to the next next slide. Okay, what is happening here now? Okay, let's do this. Okay, now in the next slide, we are now on this one that is, let me do it like this. We are still continuing with that one. Okay, the number of chromosomes in a cell is a characteristic of an organism. For example, humans have 46 chromosomes, or I will end that one, or 23 pairs. Okay, so in humans there are 23 pairs of chromosomes, which are equivalent to 46 chromosomes. Chromosomes which are single threads or single strands become double when your 2 chromatids joined by a centromere become double as a result of DNA replication. So when we look now, when we look at, let me move this one down there so that we don't mix up. Now, when we look at this one on this side, there's 2 chromosomes on this side. This one has got a single chromosome, or a single thread rather. It has got a single thread and another single thread. So here, this is just one chromatid making this chromosome and one chromatid. So it's a single stranded chromosome. All right. Once you undergo the process of what? Of DNA replication. Now you're double stranded, double means 2, single means 1. Okay, so you can see there was only 1, but here now there are how many? They are 2. Okay, now they are 2. So these are double stranded chromosome, and then that one is a singly stranded chromosome. And remember, we have said that each chromosome has got 2 chromatids. This is the sister chromatids. They are held together by what? By a centromere. Okay, and then we move to the next slide. Now the next slide, we are now looking at the differences between diploid and haploid cells. The differences between diploid and haploid cells. You need to know these terms, grade 12 learner. Xa sithetha nge-haploid cell, masiqale siyidefine so that's kakuhle ne. Haploid is the quality of a cell or organism having a single set of chromosomes. Yet diploid is when a cell or organism has paired or 2 sets of chromosomes, one from each each parent. Ne? Now, what you need to know is that, all right, a body of any organism, all right, a body of any organism has got what? Has got what we call cells, all right? Has got cells, and that's why the problem here. Has got cells. Now these cells are divided into 2, okay? Are divided into 2. Kukhona the cells. These cells, we find them on what? We find them on what? On the body. Sisibize ngoba somatic cells, these ones that we find in the body. Okanye ezinye incwadi sisibize ngoba zintoni? Body cells. All right. But also we are having another kind of what? Of cells. All right. But yona siyifumana emzimbeni wonke. Xa sithi somatic cells, thetha nge-cells ozifumana in your skin, in your bone, in your blood, in your hair. Those are somatic cells. But other kind of cell are the cells that we do not find in body kahle kahle. But these cells is in the human reproductive organs. Remember, if human reproductive organ, a testis in male, an ovary in female, now is still a body cell, is still a body part rather. So in a body cells, in a somatic cells, but with testes, kukhona izinto esizibiza ngoba seminiferous tubes. There are tubes there. Phaya kweza tubes kukhona esithi zii-germinal epithelial cells. Those the epithelium cells, they are the ones ezokhupha the cells esingezizwe somatic cells. Ezi cells ziphaya sizibiza ngoba zintoni? Zii-sex cells. All right, sizibiza ngoba zii-sex cells okanye sizibize ngoba zintoni? Zii-gametes. Right. So into ekufuneka uyazile ukuba ii-cells emzimbeni womuntu zihlukene kayi-2. Kukhona somatic cells, bekhona ii-gametes, okanye khona i-body cells, bekhona ne-sex cells. All right. Now that's into that is very important kula magama sithetha ngawo. Sithi apha sifuna ukwenza umehluko between diploid and haploid. Ii-somatic cells okanye body cells zizii-cells ezineza chromosomes besithetha ngazo sithi zii-double, ne? So we've got i-chromosomes in body cells originally ebezisiwa kumama nezinye originally ebezisiwa kutata. So we have got 2 sets of chromosomes in what? In a body cell. So xa kukhona 2 sets of chromosomes, la 2 sets of chromosomes thetha ukuthi enye isiwa kumama, enye isiwa kutata, 1 kumama, 1 kutata. So that means 2, right? Then that is called diploid. All right, that is called diploid. And then we've got the sex cells. With sex cells, we do not have 2 sets of chromosomes. We are having single set of chromosomes. Kanjani? I-body cell ezofumaneka in the seminiferous tubules izo undergo meiosis. I-meiosis siyayazi uba umsebenzi wayo ukuthini. It is to halve the number of chromosomes. So izohaflisha la diploid chromosomes imenze abe ngu-haploid. So uma ndibuze umzekelo uyayazi lapha kuba ihafu ka-4 ngu-2, ihafu ka-10 ngu-5. So kule case kuthiwa diploid. So ihafu ka-di, u-di means 2. Ihafu ka-2 ngubani? Ngu-1. So la eza sex cells ufuna zona zibe njani? Zibe haploid ngoba u-haploid is the half of diploid. In other words, a sex cell has got half the number of chromosomes as compared to the what? To the somatic cell. I hope it's clear. Now, for sexual reproduction, let me try to clear here. For sexual reproduction to take place, a haploid male gamete will fuse with a haploid female gamete during fertilization. The result is a diploid zygote. This is quite clear. Otibana, a haploid. Here is the haploid. Where is my pen now? I'm trying to use this one here. Okay. Now here is a haploid cell. It has got haploid denoting ngubani ngo small letter n, and then diploid denoting ngubani go 2n. Okay, so it's a statement again go sexual reproduction. A haploid may gamete, that is a sperm cell, is nasi paya and denoting ngabani go yellow paya. Kuthiwa is going to fuse with what? With a haploid female gamete. Where is it? And look down there. We're having a haploid male gamete. Okay, during the process of fertilization. Yepi ukuthetha ukuthi fertilizer phaya sperm cell nucleus yayo yaso ingena Excel. And then the result of that, kuzophuma ntoni ekugqibeleni? Kuzophuma a diploid zygote. All right, this is zygote because by example, sperm plus ovum is equal to zygote. Ne? So kuphuma a diploid zygote. La whole, a half, that is n, nenye half n, that is a kupa u2n, or into or you whole into etheni epheleleyo. All right, then now we move to the next slide. Now sex cells okanye gametes, that is a sperm and ova, are haploid, that is n, 23 chromosomes in human. Okay, somatic cells zona zingapi? Body cells are diploid, which is 46, that is 23 pairs of chromosome. Now la diploid 46 chromosomes, kule case humans, haploid 23 or 23 half kabani 46. So is sperm cell ne egg cell, in each of them you will find half the number of chromosomes. With males, I mean with humans, sperm cells na 23 chromosomes. With females, egg cell has got 23 chromosomes. All right. Now the 23 pairs of chromosomes that result in a zygote are divided as follows. Singena ke ngokuchromosomes, andisithethile nge-cells. Sazohlukaniswa ukhona intoni? I-sex cells nantoni? Ne-body cells. Sazinika amagama escience, sathi body cells intoni? Zii-somatic cells. And then i-sex cells, that is intoni? Zii-gametes. Now, singena ngaphakathi ke ngoku-cell A1. All right? Kule cell A1, sithatha i-cell yantoni kule case? A-body cell, the somatic cell, yona ezoba 46 chromosomes. Okay? Kweza pairs ziyi-23, 23 pairs that we're having, there are 22 pairs. Okanye masithi ezi chromosomes zihlukanisa kangaphi? Kayi-2. 1 kubani? U20 bani? U20, 22 pairs. Esizibiza bazintoni zona i-22 pairs? Sizibiza ngoba zii-autosomes. All right? Zii-chromosomes ezi. Bona eligama lapha ezantsi lithini? Let me just draw a line there. Eligama lapha ezantsi lithi somes. Yabona that suffix, I think it's a suffix ngokwesingesi, ne? And then this one again, somes, that means it's from chromosome. Okay? And then ungayi-confuse nabani? No somatic cell, nabani? No sex cell. All right, so we've got the first one, 22 pairs, esizibiza ngoba zintoni? Zii-autosomes. So xa ubuza uba how many autosomes are found in a human cell? Uzothi wena zingaphi? Wrong, ayingo-22. Uzothi wena 44, because alothetha nge-pairs. 22 pairs is equivalent to 44 autosomes. Zii-pairs eziyi-22, but i-chromosomes zona, we must put this in mind, ne? You must put this in mind, but we are talking here about the what? We are talking about the pairs here, all right? We are talking about the pairs, okay? So one pair of chromosome yaziwa ngoba yintoni yona? Yi-gonosome. I-pair A1 yaziwa ngoba yintoni? Yi-gonosome. So how many gonosomes in a human cell? Uzothola zingaphi? 2 gonosomes, but 1 pair. Okay, not 1 gonosome, because you're gonna lose serious marks for that. How is this gonosome represented then? Njengoba sithetha ngoba khona i-gonosome. Okay, yohlukile i-gonosome. I-gonosome yohluke kanjani? Kwi-females sinento esithi ngubani? Ngu-XX chromosome. What's happening here? We have got XX chromosome. That means lapha i-1 ye-gonosome sithe ziyi-2 chromosomes phaya. Enye chromosome ingu-X, enye chromosome ingubani? Ingu-X. But when it comes to the male, akufaniwa ke ngokwe-male. I-male yona inangaphi? Ina-2 nayo, but ngubani? Ngu-X and Y. All right? Ngu-X and Y. So that's how ke ngoku sizohlula khona uba oh, ezi chromosomes sizibona apha kunento sazothetha ngayo ngapha ngezantsi zezantoni? Zeze-male. Zezantoni ezi? Zeze-female. Once yaba ngu-XX, that's a female. Once yaba ngu-XY, that's a what? That's a male. All right? khona ngento kuthiwa yi-karyotype apho kuvela zonke ezi chromosomes, right? Kulapho sofike sibone khona. So wanikwa i-karyotype nje kwathiwa, look, what is this a karyotype represent? Is it a female? That is it of a female or of a male? You know, when by yantoni uzojonga ku-pair number 23. That is that one pair. Ujonga uba is it XY okanye XX now. So if it's XX, that's a female. If it's XY, that's a male. But yeah, sometimes baye bangamabhala aba-XX. Mhlambi benze something like this and something like this. Okanye bangenzi kanjalo benze something like this and something like this. All right? Now, how do you interpret these two things? You know very well. It's simple. X no X zizinto ezifanayo, ne? So once zafana, noba zimfutshane noba zinde, you know uba yintoni lena, this is a female, okay? Ngoba ziyefana, ne? You know again uba no man, u-X no Y abefana. So that means enye kwezi ngubani ingu-X, enye ngubani ingu-Y. That means this one is what? Is a male, not a female. Okay? So you must be smart like that when answering these questions. Injani le nto umfundisi othetha ngayo yegonosom what and seeing this. Let us turn to the next slide. All right? The human karyotype. That's where we're going to see what we are talking about. Human karyotype. Ufuneka ndimane ndiphosa kancinci just to drink water and then come back again. Okay? The human karyotype. Njengoba sitshilo, humans have got 46 chromosomes arranged in pairs. Each pair is different from each other by shape, size, and genetic composition. As fun as a pairs. From pair 1 to 22, that is autosomes, then 23rd pair, that is gonosomes. Okay? And then XX for male and XY for males. X is long. This is very important. This is very important. X is long. Let me put this one here. But X is long. So that means you need to know this. Uba xa udibana nento abangayibhalanga X bavele benze umgca, lena inde ingubani unge-X. Okay? And then it long and it is also big over Y. Ne? That means Y is short and small. All right? It's clean like that. Okay? Now, this is a human karyotype, this one. This is a human karyotype. Let me try to draw a circle here that shows that this is a human karyotype. Let's put it a little bit on this side. Okay. Yes, it's gonna be okay there. So we are saying this is a what? A human karyotype. Okay. This is a human karyotype. Let us also put there an arrow that's going to show what are we talking about exactly. Okay, this is a human karyotype. Now, I am going to ask you a question, and then you are going to pause a video for just 5 seconds. When you are looking at this karyotype, is this karyotype representing a female, or is this karyotype representing a male? I think you now got the answer. The correct answer here is this karyotype is of a female. Why? Because when you are looking at chromosome pair number 23, which we have said is what? Is a gonosome. You can see there that you have got an XX chromosome. You have got an XX chromosome there, as well as the children. Let me put it there. You have got a what? An XX chromosome. So XX chromosome, when we've got X and X, as it names, it's obvious that here we are talking about a female. This is a female, not a male. So you're gonna get a what? A tick for that. Ne? Why are you getting a tick for that? It's because of you got the correct answer. Ne? So you get a tick for that. So the correct answer is a female. Now, let us move on to the next slide. Now, you need also the meiosis, you need to know mitosis. But let us just stop here for now. Then on our next lesson, we are